Hello, everyone, and welcome to today's live broadcast, Adding Dimensions to Multiplex Molecular Imaging. I am Jen Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Leica Microsystems. For more information about our sponsor, please visit their site at ligomicrosystems.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenter, Dr. Scott Fraser, Elizabeth Garrett Chair of Convergent Bioscience, Provost Professor, Departments of Molecular and Computational Biology of Biomedical Engineering. For a complete biography on our presenter, please visit the Biography tab at the top of your screen. Dr. Fraser, you may now begin your presentation.
Well, thank you for including me today in the program. I'm very excited to tell you about what we've been doing in the lab to try to add dimensions to the multiplex imaging we do, mostly of live specimens. And these three examples are things that I'll draw on a bit today and try to amplify our ability to follow live multiplex images in living specimens. So I'll start by saying that one of our goals is to translate our work. And so we take the funding that we've been able to obtain from some very forward-looking foundations and federal agencies and have converted that through some patents through Caltech and USC and the Alfred Mann Institute into a number of different corporate relationships. And none of the work that I'm going to present is really conflicted today, but I just think it's wise to start out by disclosing all the possibilities. Okay, so our goal in the lab in the Translational Imaging Center has been to try to do systems level analyses in complex systems, like developing embryos. And this 21-hour embryo that you see in the upper right part of the screen is a zebrafish embryo. It's gone from an egg to an animal that has a beating heart and starting to make important neural connections in its brain in less than a day. So embryos are ideal for systems level questions because they're somewhat choreographed and things happen in a small enough size and fast enough to use imaging as a way of addressing it. Now, most people recently have been doing single cell analyses, and these single cell analyses are very powerful. They can tell us about the transcriptome or the proteome inside of the cells. But most of these analyses involve taking the cells out of their normal context. And when they're out of their normal context, we, of course, don't see their normal neighbors. We don't see the context in which they're sitting. And we also don't know what happens next or what happened before. The dynamics are really lost. So one of the issues is that these analyses show huge levels of heterogeneity and variability, which we have to try to make sense of. What we've been trying to do is to do an imaging analysis or a multiplexed living imaging that really allows us to do an embodied analysis of those cells. So now the dynamics and the variance becomes really our signal rather than the noise that we have to explain away. And of course, this is going to require tools that are tailored to this task. So what I'm going to do today is try to draw on a few different stories in the lab to give you a feeling for the ways that we're trying to do this. And some of these will be in fixed preparations, but most of them will be in living animals where we can follow the dynamics over time. So I'll return to this slide at the end, but I wanted to start by just saying that the team that's made this possible has been just a powerhouse. So I'm going to start by talking a little bit about the ways that we look at gene expression. And almost everybody in biology has done some sort of in situ hybridization to look at where RNAs are expressed. And almost everybody uses an enzyme-based approach to get enough signal above the background. You can see in the left, there's an embryo with domains that are labeled red. That red is a reaction product that an enzyme is laid down because the appreciable background of a whole-manid zebrafish embryo that's shown in gray in that image is high enough that if we just used a direct fluorochrome to label the individual mRNA by using an antisense probe, the signal would be lost in the noise. So typically an enzyme is directly or indirectly conjugated to that antisense probe, and then a chromogen is used to create a reaction product that can be seen, that amplifies the signal above the noise. The good thing about it is that it works. The challenge is to multiplex with it and to get out the sort of quantitative data that we're used to for analyses, say, of single-cell RNA sequencing because of the nonlinearity of the chemistry. So it's not really possible for us often to see the difference between subtle changes in expression because the amount of time really alters the way that things develop. So if I look in the left-hand side of this slide, you can see the normal RNA-seq sort of approach, where here a few dozen cells are looked at for a number of dozen of genes, and the code in color tells us what its expression level is. To the left is the problem we have with normal in situ analyses, where this is the same gene, PAX2, at the same stage of development, in the same strain of mouse, and in fact, in the same paper. And depending on how long the reaction was allowed to go with the chromogen, you can see that the patterns can be quite a bit different. What we wanted to do is to build something that would let us do this in a quantitative fashion. And luckily at Caltech, where we were at the time, there were people working on engineering DNA interactions. And the one that we decided to exploit is called the hybridization chain reaction. And we figured that that would allow us to do quantitative in situs. I'll show you just a little bit about that here. It's based around these two hairpins. And these hairpins are actually somewhat complementary to one another. They would really like to bind to one another, but they're trapped in this metastable state where the two halves of the hairpin are bound to one another. And so they will sit there very nicely, even though they'd like to be binding to one another, because they'd have to open up and find one another in solution before they found themselves. And that's very unlikely. The only thing that breaks that sort of situation is if an initiator sequence is present. And the initiator, which you can see is the little squiggle at the bottom, has a sequence that's designated as A star B star. That means it's complementary to the A that's in the toehold for hairpin 1. And so if it binds to that toehold, now it can make base pairing to the other half of the hairpin, displacing it, doing a strand displacement that opens up that hairpin, and it reveals C star B star, which happens to be the initiator for the other hairpin. And so what we end up with now is something that regenerates A star B star, and that can grow explosively. It grows explosively to about a 200 mer, and we can see that simply in gels here, where we're running hairpins through an electrophoretic gel. If the hairpins remain as individual hairpins, they run to the bottom of the gel in the electric field. And I've got this gel with 4 different pairs of hairpins labeled with different dyes. The star in the picture to the left represents dye molecules. And you can see that they migrate down at the bottom of the gel, even if I add a blender extract of whole zebrafish. So there's a lot of RNAs around, and it doesn't cause the amplification. If instead I add initiator 1, you can see that blue disappears from the band at the bottom and appears up above. And it's about a 200 hairpins that have assembled to give that band. If I add initiator 3, that's the initiator for the green hairpins, and you can see that green disappears from the bottom and it appears again as a 200-mer at the top. So this gives us a chance to give us ourselves a defined amplification. The initiator gets turned into about 200 dye molecules. To make this into an in situ hybridization approach, we make an antisense probe. This antisense probe hybridizes to the to the RNA that's in the specimen, and it has initiator sequence hanging off of it. So when the hairpins are added, they amplify. They amplify in a way that the RNA is tagged not by one dye molecule, but by 200 per initiator. What this allows us to do is to use small reagents that assemble in situ and therefore can penetrate well throughout specimens. So we find that the reaction product amount is proportional to the amount of RNA that's there, independent of whether it's near the surface or deep within. We also find nice linear gain, which means that unlike conventional in situs, where dim things are often lost, you can see the nascent transcripts that are being generated in the individual nuclei are lost in the conventional in situ on the right. If we look at the HCR just below that, you can see that the dots in the nuclei capture the dim, relatively dim and nascent transcripts that are in the nuclei. A few years ago, V. Castrovetti worked on characterizing the linearity of this technique, and one of the approaches he used is by tagging the same RNA with a couple different colors so we could compare it. And two important things are shown in this plot. One is that the dimmest signal is above 0. So it's easy to tell signal from noise. As you can see in the pictures, you don't see that indeterminate gray in the background anymore because the signal has been amplified above that noise. The other thing you can see is that we get a nice slope of 1 where the data clusters very nicely. So it means that we've got a way of linearly recognizing the RNA, able to see even low levels because the 0 is clearly at above 0, and we're very happy with it. In fact, the quality of the signal is good to just a few percent, well better than 5%. Now, one thing that'll keep on coming up today is that, biological imaging really has no free lunches, and we're always trying to trade off photo damage against speed, against resolution, and how deep we're going in the specimen. And so one of our limitations in any imaging study then is to figure out how to get enough signal per unit time that we can image more quickly. And so one of the ways that we've been trying to do this is to add a system to the HCR. In this case, we call it Muse. It's been done by a couple of my senior colleagues in the lab. And this allows us to get another couple orders of magnitude in gain. So now what we're doing is doing the same amplification, but adding to it another round of linear amplification. This gives us the ability to put that initiator sequence on antibodies, for example, shown on the left top, or on RNAs, shown on the right top, or even in advanced sort of things like CyTOF labels, so we could do imaging mass spec as shown at the bottom. So this has allowed us then to put different initiators on different antibodies of the same isotypes. In this case, it's fourplex with all rabbit antibodies to the left, or to put 5 different initiators on 5 different RNA molecules. You can see the individual dots that represent the individual messages. And if we blew this up, you'd see that they're very good about being orthogonal to one another. That is, that the dyes are very localized to their own RNA and don't cross-react. Or we can do, for example, in this case, a 5-plex, where we're doing in situ hybridization for 3 RNAs plus immunofluorescence. So this technique gives us a little bit more signal and lets us go faster. Now that's good news. It means that we've got a technique where we can do, a half-dozen plex at least. Long Kai at Caltech has carried these sorts of approaches in serial staining to give him, hundreds to thousands of different genes. We can look at something like a mouse embryo shown here with multiple different genes in a way that the staining is independent of how long the animal stained for and how long it sat in the fridge. So in this case, it sat for a month or 2 in the fridge before it was imaged. And so this gives you a chance to do quantitative in situs without having to worry about how to compare the results. That's the good news. Now, the not so good news is that that was 5 gigavoxels of confocal imaging. And that means that we've got a lot of imaging time. In fact, that was 10 hours of imaging to collect this mosaic image stack of 4x6 image stacks. And in fact, you can see just a hair, if you've got sharp eyes, of where the image was scanned twice, where there's a little bit of extra bleaching. And so this limits our ability to be quantitative, and it also limits the time. And of course, what we'd like to do is image at much higher power to look at the individual dots I showed you before for the Muse reaction product. For doing HCR or Muse or RNAscope or Stellaris or any of the other fluorescence in situ techniques, this would have to be much slower. So that 10 hours would be an impractical amount of time if we needed to go at high power. So that's one limit. The other limit is what we'd really like is to be able to tag quite a bit more per imaging session. So we'd like a larger number of colors. Let me just address the speed issue quickly. I mean, I mentioned this trade-off earlier. If we want to go faster, we normally have the problem that we add a lot more photo damage or need to sacrifice resolution or sacrifice depth. What we've been trying to do to reach a new balance or new compromise is to adopt the light sheet approaches that many others have been using in the last few decades. It's a technique invented about a century ago for looking at colloidal solutions, but then really reinvented a few times over the last few decades. And the most popular one is the one that Ernst Stelzer and Jan Huchsen and their co-workers did, where light's brought in from the side and then the fluorescence is collected in parallel. Our version of this moves to 2-photon illumination, and we do that because for the reason shown in the lower right. You can see as a laser beam moves through the screen from right to left, that as that light is focused, it gives you almost an hourglass shape of beads in a gel that I'm imaging here with 488 nanometer excitation. If instead we use 2-photon excitation, as we look to the left or the right, the photons aren't concentrated enough to give an excitation, so we get a relatively thin light sheet that we can position even deep into a specimen and collect a more accurate light sheet within the specimen by moving this excited beam into and out of the screen, for example. This has allowed us to do 2-photon spin on a variety of different specimens, and I just show one here, which is a fly embryo. We use them a great bit because they're dramatically more sensitive to photons than most of the other specimens we use. So we're able to see if we can image without perturbing by using the fly embryo. And you can see it hatched and crawled away. In fact, that crawling away happened at the same time as it would have if they were hatching on their own off the microscope. Here's imaging within the brain of an intact animal. We've been able to image for up to days to be able to follow the activity, in this case, followed by using GCAMP6S, a calcium indicator that's genetically encoded. And you can see the individual flashes that are the cells as they become active and their calcium levels increase. So we've been really enjoying the 2-photon light sheet. It's a way to compress down the imaging time more than a couple orders of magnitude. And it really allows us to jump onto the Moore's law of camera development, so that the faster, better, bigger cameras that are being developed can be used to see larger fields of view faster and with better sensitivity. So what about the color issue? Well, the color issue is one that has haunted all of us, because almost every fluorochrome that we want to deal with has a pretty broad emission spectrum. Here I have a cartoon of a zebrafish embryo in which the vasculature is labeled with EGFP, and the muscle cells through which that vasculature is running is labeled with citrine. You can see the spectra overlap a great bit, and if we look at the true color, you can't see those as two distinct things. So what we want to be able to do is to separate out these overlapping fluorochromes and also separate it out from the many other things that are there. Now, one temptation would be just to use very narrow bandpass filters, but as you can see, even with this simple cartoon, it would be very hard to find one narrow bandpass that would reject one of the fluorescent proteins and collect light from the other one. To get around this problem, we were able to adopt the approaches that have been developed by the remote sensing community, the people that fly satellites and interpret their images, to collect an image cube. In this case, the image cube is X, Y, and lambda as the third dimension, by adding a spectrometer to the confocal microscope. One of the early adopters of this approach under license from Caltech was the Zeiss multispectral imagers, as the one shown in the lower left. And this shows you some early prototype results even that worked really well. Even 8 spectral bands within that narrow green region allowed us to separate out GFP from fluorescein, even though no one of those images does, and even though the spectra overlap tremendously. You can see here the separation out of the GFP from the fluorescein, but you can also see one of the weak points. And if you use sort of eagle eyes, you might be able to see that in the region of red, that's the phalloidin labeled with FITC that's been pseudocolored red, then the region that the nucleus is, the noise in the GFP image is being imposed on the fluorescein image. So you see a little bit of speckling there. So in dim things, you're dominated by the worst signal to noise in the image cube. And this has limited us to the ability to go after multiple colors at once. Luckily, representatives from Enrico Grattan's lab moved to my lab to start doing some research and applied the Fraser approach to this. The Fraser approach, for those of you that aren't familiar, is to take the Fourier transform of the spectrum. So if we look here on this region of the animal where there's only green dyes, we would get a green spectrum as shown at the top of the spectral plane. If we looked in places that are only red, we'd get a red spectrum. And if we take the Fourier transform of the green and the red, what we can see is that the green turns into a green dot in the Fraser plot. The Fraser plot is the sine and the cosine of one of the harmonics of the Fourier transform series. What's convenient about this is that a 50-50 mix is a dot halfway in between the two, and a 70-30 would be 70% along the line. So basically it takes something that involved before inverting a matrix into a forward problem, and this forward problem both is dramatically faster. Here I'm comparing, trying to separate out the signals using optical filters, trying to do linear unmixing, and finally trying to do hyperspectral Fraser. And I think what you can see is that the bottom separation is much better for getting a high signal to noise rendering of the vasculature and separating it from the muscle cells. It also does it quite a bit faster. So if we just compare then these results in a live animal, well, the previous was as well, but going through several different indicators, in this case, blue fluorescent protein, yellow and green, and also some of the biological autofluorescence, the xanthophores and the autofluorescence from metabolism and other things, you can see that we can separate out pretty nicely these dyes that overlap with one another tremendously. What would have been hours of processing for these multi-giga voxel images becomes something that's relatively fast, almost as fast as you can read the data off the hard disk. We can process it. So that solves a bit of our problem, but we still have a problem. I'm going to go back here. We still have a problem in separating these out, that these spectra are collected with a large number of photons, and we want to be able to do the imaging at a minimal light dose. So one of our challenges when we start doing that is that the spectra at low signal to noise, instead of looking like the ground truth spectra shown in this cartoon to the left, end up, once we deal with the stochastics of the photon emission from the fluorescent dyes, end up being a lot messier. And so one of our challenges has been how to use multispectral approaches when the stochastics of the dye start entering in. Luckily, folks in the lab have been applying machine learning approaches using the, in this case, synthetic data, ultra low signal to noise spectra, and comparing it to what the real spectra is, we can get a way of reconstituting the real spectra out of it and get almost the same data. This shows some real, instead of a cartoon, some real examples of this. And then a biological example shown here is an image of a doubly labeled specimen, imaged with very few photons per pixel. And you can see that the linear unmixing really falls apart at this point because we're starved for photons. If instead we use the hybrid unmixing, this is using the machine learning and the hyperspectral phaser, you can see that we get a signal to noise that's much more interpretable. So this approach is allowing us to look at the things that are most light sensitive, like growing vasculature within an embryo, and look at multiple different labels and also the autofluorescence and background noise. Now this autofluorescence has a number of different origins, and you can see that some of it is biological, like the xanthophores, they're pigment cells. Others that are things like the retinoids that are in the tissue, and also NADH, one of the energetic intermediates that can be either in a bound or a free state, depending on if the cell is oxidative or in, oxphos or glycolytic. So we can use this machine learning to sort of help us with this compromise, but and we can almost minimize the cost of the lunch, but we really want to see if we can push this further. One way to try to push it further is to deal with the problem that is always tempting with multispectral, is we see companies coming out with detectors that separate out the light into not just 32 channels, but even into hundreds of channels. And while this sounds like a very good thing, what you're doing is adding 100 channels worth of noise and splitting up the signal into 100 different channels. So you're both decreasing the signal per channel and increasing the noise in the overall thing. So one of the things we've been working on over the years with the hyperspectral phaser is to see just how few channels we can deal with. This is showing a comparison between 32 bands and 4 bands, and it's pretty notable how good the 4 bands does, even with this simple multi-labeled specimen. If we look at higher power, you can see that the individual features can be separated out almost as well, maybe as well as they can in the 32 band. And finally, if we look at it quantitatively by scanning a line, we can see that the signal to noise for the 4 channels is virtually superimposable with the 32 channel. So what this is allowing us to do now is to think about applying machine learning and fewer channels and the hybrid and mixing, the hyperspectral phaser tools, to try to separate things out. This just shows a little bit of our attempts to separate out both the intrinsic signal and the extrinsic signal. In this case, the extrinsic signals are the fluorescent proteins. The intrinsic are these different sources of autofluorescence. And you can see their position on the hyperspectral phaser plot. And while this is encouraging, there's a problem. And the problem is that collecting the amount of light uniformly is a challenge in intensity. And it's a much lower challenge in lifetime. And so what I want to spend a little bit of time now talking about is our attempt to do lifetime imaging and to combine lifetime and spectral imaging to get a better understanding of overlapping labels within the same specimen. So this hybrid around the lab we call Spectroflim. I should mention that this sort of emerged in the lab through a detente that was created by 2 alumni of Enrico Grattan's lab. Mimo Arnasano was a very big believer in lifetime imaging. Francesco was a very big believer in hyperspectral imaging. And they came to bring their forces together by, while they were at USC, by joining the 2 of these together. Just for a moment to sort of digress into lifetimes, and some people might not be as familiar with it. Lifetime involves trying to measure the time a fluorochrome spends in the excited state before it drops back down to towards the ground state and emits a photon. So the typical way that that can be measured is to use a pulsed laser and then measure the time and add those up to obtain the exponential decay that you can see in the right-hand panel. So what Enrico Grattan and his colleagues have shown is the amazing power of moving away from just doing the tau curve fitting on the lifetime curve to moving it with the phaser approach into the Fourier transform of phaser space. You can see that different lifetimes now appear at different places on the phaser plot. And if we think about the hyperspectral responses of those exogenous and endogenous labels, you can see that they show up at characteristic places. Neither one of them is really perfect, but both of them are extremely informative. And so we've been involved in trying to work with vendors and with some of our own soldering irons and such to build instrumentation that can really collect the spectra and the lifetime. And been, for example, in one experiment, shooting that down a bronchoscope so that we can see the response of the airways in both hyperspectral and lifetime imaging. The lifetime imaging is very convenient for looking at the metabolic profile. The NADH signal, when it's free, represents the more oxidative phosphorylation sort of response of the cells. The glycolytic is when the NADH is free, and you can see in this color-coded image of individual cells in the airway that we can tell the difference on a cell-by-cell basis. If we look at hyperspectral, you can see some things that we're just blind to in the lifetime, like the autofluorescence from the elastin and collagen that's shown in green to the left of the hyperspectral image. And you can imagine that combining the two is going to be the most informative. And we're trying to do that now to generate something that could be used in the very noisy environment of imaging human airways. Also been extending this to looking at organoids and really trying to see how far we can push these different modalities. And this is a picture from a paper that Mimo generated now a couple of years ago, combining together everything from lifetime imaging to hyperspectral imaging, and then techniques like phase contrast and OCT as well. So what we've been trying to do is come up with instrumentation that combine all of these, and the challenge has been to make them robust. We've been lucky as we've started to carry this to fewer and fewer cells, that the Leica, Fortune, and Falcon approach has allowed us to do this much more conveniently. So the Fortune gives us 4 different spectral bands. I've shown you earlier that 4 bands is enough to get very nice hyperspectral results out of it. And that's allowed us to do things like image over a day-long period as a group of cells that were isolated at 0 hours, all because they were stem cells. It turns out that the yellow reflects the metabolic state that's indicative of the stem cells. You can see of the 4 cells we're following that 3 of them are still a little bit in that stem cell state. One's really quite a bit out of it. A better part of a day later, 20 hours into the recording, you can see that there's only 1 of them. And then at the end, you can see that basically all of them have given up on the stem cell-like metabolism. We're now using this in culture plates, in organoids, and in intact systems to try to eavesdrop on the biology of the cells and the physiology of the cells. And while it's early days, working with the Falcon has been really powerful for getting both the spectral and the lifetime data out. So I'm going to stop here because I think I could go on and on about our children and the way we're trying to use them in the lab, our children being our different techniques and our different instruments. What I've tried to share with you today is our approach to try to add dimensions to the imaging. I showed you a little bit about our multiplex imaging and both the abilities and challenges there. I showed you a little bit about the spectral phaser analysis and showed you our ability to separate out multiple labels, even if we're only looking at 4 spectral bands. And then at the end, I showed you the ability to use lifetime and spectra in sort of doing spectrofilm imaging. And this brings up the power of jointly refining instrumentation. It's turned out to be much easier for us to support users by having integrated instrumentation that can do both the spectral data collection and the lifetime data. It allows us to jointly define what the optimal signal to noise is, like the number of channels that's really appropriate. And one of the things that we're finding as we're doing this joint development of instrumentation that can do lifetime and spectra is that less might really be more. And we're still trying to find the right compromise, but it might really be that as few as 4 spectral bands is enough to give us the best low light level performance and also very usable label separation. So I'm going to end by thanking the people that really made these technologies possible. The multiplex in situ drew on a collaboration with Niles Pierce and his colleagues at Caltech when we were there. Lei Trin and Vikas Trivedi were the real driving forces on moving that collaboration forward. And then in the further refinement of the technology that I mentioned as Muse, it was really Simon and Joe that drove that forward. The hyperspectral phaser really owes its creation to Francesco Trotrale and all of his colleagues who've been working on different aspects of improving the performance and the speed and of the hyperspectral phaser approaches and integrating them in with lifetime tools as well. The 2-photon spin's really been driven by Tai and his colleagues. And I've listed a few of them here, but of course, in any one of these lists, there's always more that we could list. And finally, the spectrofilm has really been something that has involved many labs. And as we've started to show the technologies, we've found people in ophthalmology and cancer biology and beta cell biology and a variety of other systems, these collaborating labs shown at the bottom, that are working together with the people in our lab to really refine together these spectrofilm sort of techniques. And I think I'll stop there and see if there's any questions, and hopefully you've enjoyed what I've shared with you today.
Thank you, Dr. Fraser, for your informative presentation, and we want to thank our audience for their interesting questions. All questions that come in today and those submitted during the on-demand period will be answered by Dr. Fraser via the email address you provided at the time of registration. We would like to once again thank Dr. Fraser for his time today and his important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, goodbye.